and today in this video we're going to discuss about the bacteriophage, structure of bacteriophage. So first of all, let me tell you the bacteriophage is basically the virus which infect only the bacteria. So these are basically the group of virus which infect the bacteria. Okay, these are the virus which infect the bacteria. Now they are the complex viruses, and they were, we can say, this term bacteriophage virus was we can say coined by D. Harary in 1917. This term was coined by D. Harary in 1917. Now just remember, there were the two scientists who worked on this bacteriophage virus, and they have discovered, we can say, they have discovered this bacteriophage virus independently. Now the name of the scientist was, number one was the Towert, which worked in 1915, and he worked in the UK. Okay, another was, we have D. Harary, and this D. Harary in 1917, he worked in the France. He worked in France. Fine, they both work independently. However, the term that was coined by D. Harary virus. Okay, now we should move forward. Okay, yep. This virus is basically composed of many viruses, such as this have T phages, comprised of T phages, which includes T even phage and include T odd phage. Another here we have lambda phage. Fine. So if I to talk about T even phase, so here we have T2, T4, T6. These are even, and they are basically non-enveloped double standard DNA non-enveloped. Sorry, from this only T4 is non-enveloped and double standard DNA. Fine. And if I to talk about the old viruses, so here we have T3, T1, T7. So these are old viruses. Now let me tell you what is the difference between T phage and lambda phage. So here, if I to talk about T phage, so T phage basically this is the virus, like the virus which comprised of the fibers which have tail fibers. Okay, which have tail fibers, and another we have lambda phage. So this is basically another type of virus which do not comprised, or we can say which is not composed of tail fibers, and we can say there is a less developed tail fibers. So these are known as lambda phage, which have less developed tail fibers. Fine, and now let's see the bacteriophage virus even in more detail. Okay, so these bacteriophage viruses are basically highly host specific virus. They only kill, we can say, highly host specific. They are highly host specific. Another thing, they can only infect, we can say, single species, or they can infect the strains within the species. Now they can, we can say, yeah, they can infect single species, or can infect strains, various strains within the species. Now this, we can say, a species contains some strains, so they can only infect a single species, or they can infect the different strains which are within the species, okay, which are in that species. Fine, and now let's see the bacteriophage virus can be enveloped or can be non-enveloped. This can be enveloped or non-enveloped. can be enveloped or non-enveloped or non-enveloped. Now let's see the discovery. As I told you, the discovery was done by two scientists independently. So now what they have, we can say their observation of these two scientists. So their observation was basically the bacteria colonies are suddenly undergoing into lysis. They both find out the observation that जो bacteria की colonies होती हैं, they are undergoing into the lysis, means suddenly they are baking. So they found that bacterial colonies suddenly are undergoing suddenly are undergoing into lysis. Into lysis. Fine. So now we are going to discuss about the structure of bacteriophage. So now let's see the structure of bacteriophage. Here I'm going to show the diagram with you guys. And I have shown the diagram here on this screen. So now let me tell you the structure of this bacteriophage. Okay, so in this structure, basically there is the complexity of viruses basically depend on the structure, and the bacteriophage virus composed of head, tail, and neck. All three are included: head, tail, and neck. So now first we have what? First we have head. So basically the bacteriophage head is polyhedral, or their capsomeres are arranged into the isohedral manner. When their capsomeres are arranged into the isohedral manner and their head is known as polyhedral. Polyhedral capsomeres arrange in isohedral manner. Fine. Now this isohedral manner, here we have what? Here it will contain 20, we can say, 20, we can say, not, yeah, here we have 20 phages, and it include 30 ages. These 20 phages include 30 ages. Okay. Another thing we have is the genome. There we can say, yep, the phage consists of what? With genome? Double standard DNA linear genome. They have double standard DNA linear genome, and we can say this is basically the in specific term, which is deoxyribonucleic acid. It includes deoxyribonucleic acid. Fine. And let me tell you, when capsid, we have capsid plus we can say genome, or we can say nucleic acid. So it will form what? Nucleogenome. Capsid, nucleic acid, sorry. Nucleocapsid. Fine. So here furthermore we have basically, yes. Moreover, here we have neck. Now we are going to see the neck. So the neck is basically, here we have short tube neck, which is made up of what? Proteinous. Their structure is made up of proteinous structure. Proteinous structure. And this neck help us to join the head and tail of the virus. So here you can see, if I can show you, this is the head of the virus and this is the tail. So here we have, this is a neck which help us to join the head and tail of the virus. Fine. Help in joining head and tail. It joins head and tail. And here neck also consist of, we can say here, collar. Collar is also present. It comprised of collar. It comprised of collar. Okay, I hope this neck is also clear to you guys. Now we should go with the tail. Tail. So if I can give you, so bacteriophage tail is basically composed of various proteins, and this tail help us to, we can say, this is the, from the neck region, this tail is basically hollow tube, and this is why this is hollow, so that we can say the genome which we have injected from the outside, so that can go easily by this hollow tube. So injected hollow tube intended it is, so that this is basically we can say channel of genome injection, or we can say other covering of hollow tube. Okay, channel of genome, so that genome can pass easily, which genome is injected. Now here this tail, basically outer part of the tail, here we have what, the contractile. We can say here we have hollow tube which is contractile. Okay, and we can say here we have tail sheet, like contractile tail sheet. Okay, and which we can say allow genome to go, we can say, further downward. So this tail is, we can say, contracted. So here as you can see in this diagram, first we have what, this is the tail, and this is what, when this is pinning. Here this is another host, we can say, body where it is pinned. Now when it is pinned, so here you tell contraction and penetration. So this, here you can see the genome is what, here we have genome, and genome is entering inside this hollow tube, and this is what this tail sheet, or you can say contracted is. This is, you can say, contracted, and when this is contracted, so genome is going further inside. Okay, yeah. So this is what contractile sheet. Another thing we have what, tail fibers. So now there's the big role of tail fibers. So tail fibers are basically, they are six in numbers. And they attach to the end plate. Like it provides a strong attachment of the, we can say, tails with the host. Like they attach with host cell. Okay, now how they attach, as these tail fibers contain what, the receptors which are binding protein, and they are attached to the host receptor site. They also, this contain receptors, and they attach to the host receptor site, to host receptor site. Fine, now last concept we have of base plate. So let's see here the base plate. Base plate. Fine. So base plate is also known as what? This base plate is also known as end plate, and the shape of this base plate is basically saddle shape, or you can say base plate composed of, or comprised of tail fibers. Tail fibers are present, and here we have pins are present, and their pins are basically, pins are retractile. Their pins are retractile. Now what does this word retractile means? Means they can, we can say, attach to the host receptors, and they can easily come out outside too. मतलब ये attach होते हैं फिर वो बाहर easily निकल भी सकती हैं. So their pins are retractile when they attach, and their pins, what, their pins possess an enzyme which is lysozyme. Possess the enzyme which is lysozyme. And what is the work of this lysozyme? So basically the work of this lysosome basically create the tiny holes into the, in the wall of bacteria, in the host cell or bacteria. As their host cell is what? Their host cell is bacteria. So lysozyme create tiny holes in the bacteria. Tiny holes in bacteria. So this is how, from which the genome which is injected, it can go directly into the bacteria. So I hope that with the help of this structure and diagram, it is clear to you guys. So in the next video, we're going to discuss about the life cycle of bacteriophage. Till that, bye bye.